science research with the University of Alaska Fairbanks. And I'm a soil scientist. I study soils, soil biology and ecology in agricultural soils across Alaska. And I'm stationed in Palmer at the Matnuska Experiment Farm Extension Center. Today we're at the Tyonek Garden, and we're going to be sampling soil as part of a statewide soil health survey. The health survey that we're doing is an effort to just really understand the nature of agricultural soils across Alaska's different regions, different growing conditions, different cropping systems. It started in 2023. This is our third year. And this year we're doing a lot of microbial analysis, so that's what's new. We're just trying to understand kind of the baseline condition, what kind of microbes live in our soils, what do they do, how active are they, and how do they differ across different regions within Alaska. To me, healthy soil is soil that supports life, including plants. As a soil biologist, I think a lot about all the different critters that live in the soil. Many of them are helpful to the plants that we want to grow. And so I think about a healthy soil as like a healthy habitat, which means that it has food for all the critters that live in the soil, including the plants, lots of nutrients. It has the ability to hold water, but it doesn't hold water to the point where it gets soggy. So the water drains, and then the pore spaces in the soil hold enough water so that the plants can get what they need without being flooded. A healthy soil just provides all of those kind of critical pieces of growing environment and food that a plant needs. So plants need air, water, nutrients. They need freedom from disturbance and chemical toxins, and a healthy soil has all those things. Yeah, so erosion is, it's kind of like a bleeding artery in terms of soil health. That's like the human health analogy, which means that it's a critical loss of soil. If soil is eroding, that's the loss of nutrients, it's the loss of organic matter, and nothing else matters in that instance because the soil's in an active state of degradation. And so all of the things that we do to create a healthy soil prevent erosion. Usually we just try to keep the soil covered with as much material as possible and then minimize the risk, the exposure to the elements. So exposure to wind and runoff. So keeping mulch on the soil, keeping things like windbreaks around the garden, and then always living plants that have roots that are anchoring the soil down. Those are good practices. A healthy soil is very strong, and the soil particles are held together with roots and fungi, and there's like glues in the soil that keep the soil binding to itself. And so a healthy soil is much more resistant to erosion, and it kind of perpetuates that stability over time.
Cool.
The tricky thing about soil science is that there's very little that we can see from the eye in terms of what a plant needs. So we need to rely on lab tests and chemical extractions or microscopic methods. And so today we're taking soil samples. We'll take the samples back to the lab and have them analyzed for all the different properties that tell us something about its health. So we'll take two different kinds of samples. One will be what I call a bulk sample, or just a loose soil sample. So I have a trowel. I like a mason's trowel. And I'm going to try to represent this area that we're sampling. So I'll take maybe 3 or 4 different small scoops and mix them in the bucket, and then I'll portion them off into the different baggies and take them back to the lab. And then the samples that go into the bucket, those get portioned off. Some of them are analyzed for microbial biomass. Some are analyzed for all the different chemical properties like pH, organic matter, nitrogen, phosphorus, potassium. And then we have some that we're just gonna archive away in a freezer, and someday maybe we'll have the ability or the funding to look at the actual microbial DNA in those samples. And then we also have this special coring device. It's called a hammer core, and we use a lot of weight. It's very heavy, and we use the weight to drive the cylinder into the ground and then remove a core that maintains the structure of that soil so that we get a better idea of how much pore space versus particle space is occupied in a given volume of the soil. So the sample from the hammer core just tells us basically the physical arrangement of the soil particles, and that's it. It's just a mass divided by a volume. So here at the Tyonek Garden, we took samples from some of the beds that have been prepared for growing veggies, like brassicas, and they have been treated with compost and formed into the bed. And so we sampled those. We also sampled a field that's being prepared for potatoes. And the potatoes haven't been planted yet, but they will add some compost and then build the potato hills. And then we sampled an adjacent area that is natural vegetation, and that's, the assumption there is that that's what the soil looked like before we developed a garden here. And so it's always a really cool comparison to see what the natural state of the soil is in comparison to what it's been, what it is after it's been managed. A big one for Alaska is adjusting pH. So a lot of our soils have really low pH values. That's just because of the natural properties of our soils here. And so oftentimes that's an annual amendment that we need to kind of continue to check up on. And so if the pH is too low, then we can calculate lime application rates. And so a farmer can apply liming material to increase the pH, make nutrients available, and minimize aluminum toxicity, which is a problem with low pH. I recommend for a home gardener or a farmer, you don't have to sample every single field or bed every year unless you are changing something in your practice and you want to track those changes. But for example, a farm where you have multiple fields, maybe each year you sample 3 fields and then you kind of rotate that sampling through your operation so that every year you're sampling soil, but maybe not every field every year. Sampling soil on a 3 to 5 year time scale is usually sufficient. We typically aim to have the test results back to our participants by Thanksgiving, so late November. We have fewer participants this year, so it may be earlier in the fall. But the idea is that then after the growing season, when the growing season is still kind of fresh in everyone's mind, they can look at the report and start to make plans for next year. If they need to buy materials like lime or fertilizers or different amendments, or if they want to just see something in the test and decide over the winter that they want to try something new, then we have time to plan and see.